just wanted to get started with a quick description about our experience so far using Promethean instrumentation here at Mayo Clinic. And our focus today is going to be on a couple of medically relevant regions. But I just wanted to disclose that we do have a business agreement. We get discounted supplies and reagents and a lot of scientific and technical support provided by ONT. In particular, I wanted to call out Andrew, Daniel, Maddie, and Mel as being the individuals that have helped us stand up what we're doing today and what we'll have the opportunity to describe. Without that, I think it's almost impossible for me to give this talk today. So I'm from Rochester, Minnesota. It's a quite snowy place, isolated in an area of the United States that's flyover country. Our medical clinic is a foundation that does have a position in the middle of the United States, but we have clinics in Arizona, Florida, and we're a health system. And you'll hear a little bit about the integration of genomic testing in the health system tomorrow from one of my division chair, Dr. Kip. But we also have a global presence. We even have a clinic here in London that I've never visited, thank goodness, but also a lot of collaborative hospitals throughout the world. We're really focused in a variety of different laboratory support services and reference laboratory activities in the clinical space. One area of concern and focus for us has been inherited disorder testing. An area that is massively underutilized, I think, in the clinical space is the capacity to query areas of the genome that are very difficult to assess. And it's hard to come to a place where everyone is very familiar with long-read sequencing and then talk about dark genomic spaces, because I think those two often are incoherent to each other. But I'm coming from a laboratory that does a lot of short-read sequencing and have done Sanger sequencing for many decades. And so from our perspective, there is dark genomic regions and camouflage regions that are areas of the genome that are not being met in the clinical laboratory. And there was a nice paper by one of our colleagues here, Mark Ebert, that described this complex problem, essentially that due to the short-read sequencing methodology, some regions are essentially having too much GC-rich content that can't be sequenced in sequencing by synthesis methods. We also have regions of the genome that are just dark by mapping, essentially being camouflaged or mismapped or poor mapping quality scores. And I think these regions even impact some of the long-read sequencing data. But then, of course, we've heard about tandem repeats or expansion alleles. In our area that I'll talk a little bit more in detail today will be the gene duplications and or pseudogene interferences, which can either be in tandem or more distal regions. And one in particular, an area of focus of mine has been the spinal muscular atrophy related chromosome 5 conditions, which are often due to either deletions or loss of function mutations in the SMN1 gene. And this is a nice summary of what is the observation that one sees when using short-read sequencing, essentially that there are no reads mapped to SMN1 because of a pseudogene of SMN2. How does this impact the clinic? You can see on the far right side, this is the ClinGen experience of all the laboratories worldwide that are describing their variants and their point mutations in the SMN1 gene. There is no experience. So laboratories performing this type of testing, essentially looking at every single variant that you find, if it's not in the literature, is a variant of unknown significance, and we really don't know what any clinical lab experience is right now because there is no repository for it, because there's no clinical testing that have been organized from this due to the gaps that we see in short-read sequencing. How does spinal muscular atrophy arise from this condition? It's a little bit complex, but there are two genes that describe the spinal muscular atrophy gene SMN1. This is the gene that most of the proteins expressed in our neurons from that particular loci. But there's the pseudogene, the SMN2 gene. They are almost identical. They're essentially just 5 nucleotides. There probably are more nucleotides, but we're classically considered 5 nucleotides that can resolve these two regions on chromosome 5. SMN2 has a T, an exon 7, that essentially leads to skipping of that exon 7, nonsense-mediated decay, loss of mRNA, essentially reduced expression off that allele. But we have at least, thankfully, 2 copies of SMN1 in our genome and are capable of making sufficient amount of protein from that because there is no skipping. Spinal muscular atrophy, unfortunately, is caused often by deletions or loss of the SMN1 gene inherited in autosomal recessive fashion. And what this does for individuals that inherit 2 dysfunctional copies from their parents, essentially, is that increased risk for expressing with a very progressive and unfortunate peripheral neuropathy and a loss of neuronal function that isn't reversible So these kids can present in several different phenotypes, and one of which is type 1, essentially, where before 6 months of age, they're essentially recognized as being unable to sit, having respiratory distress, and very few of them live beyond the age of 2. These individuals actually have 1 or 2 copies of the pseudogene, SMN2. The other type 2s, though, can essentially live a little bit further, but are unable to walk or stand, and many of them are alive up to the age of 25, and they'll have 2 to 4 copies of the SMN2 gene. But there's also later onset presentations that are correlated with the copy number of SMN2. And you can see our clinical experience in the figure on the far top right side over there, where our observation has been that patients that come to our clinic for a diagnosis, very many of them between the ages of 2 to 18 will have either 1 copy, 2 copy, 3 copies, or 4 copies of SMN2, but yet have a clinical presentation. So the copy numbers of SMN2 don't necessarily directly correlate with your presentation. And we essentially need to investigate this further, and there are some hypotheses behind that. There might be SMN2 variants that are associated with productive effects of that particular gene expression and allow the patients to be to live a little bit longer or present a little bit later. One of the things that we also identified through carrier testing, and here we've done about 24,600 individuals, is that we have different numbers of SMN1 copies in individuals and in different numbers of SMN2 copies. We've seen between 1 and 5 copies of SMN1, and we've seen essentially between 1 and I believe up to 6 copies of SMN2. So essentially right now in carrier testing for the deletion of SMN1, you will essentially get an allele or some form of counting for the SMN1 variant, and you'll essentially identify whether or not you have 1, 2, 3, 4, 5, 6, or etc. copies of SMN1. We will likely do the observation that we've seen people with 5 copies of SMN1 have patients who are out in the quote wild with 3 copies of SMN1 on one allele and 2 copies on the other, or a combination thereof. So there are potentially shortcomings to looking at carrier testing just using allele count as an approach. Luckily for the patients that are diagnosed early before they have symptoms, and there are a lot of newborn screening programs for SMA right now, there are therapeutic options for these individuals, but they're extremely expensive. This used to be, and I think it may still be, the most expensive medicine in the United States right now. The gene therapy in particular was about $1 million. But you can see here that there is antisense oligonucleotide that essentially enhance the SMN2 expression using an mRNA splicing modifier. But the dose delivery isn't very comfortable. If you can imagine, you get an intrathecal bolus on a monthly basis, essentially, and that has continued throughout your lifespan. We really don't know what the long-term effects are from the RNA therapy, but at this point, we observe kids who essentially have had that therapy for the last couple of years and don't need a second dose of that particular adenovirus vector. So, at this point, there is a lot of interest in trying to diagnose this condition in particular, and also characterize patients pretty quickly so that we can identify whether or not they can postpone that treatment later in life. Currently, right now, it's very difficult to do this by short read. In fact, like I mentioned, it's impossible. It's also very difficult to do this with long-read sequencing methodologies that have 13.5 kb, for instance, here, and you can appreciate in the very bottom right there, SMN1, SMN2 were not covered in the paper published using this from another long-read chemistry instrument. So we went to ONT. They were kind enough to embrace us, to help us with this problem. We also have a very astute group of bioinformaticians that have set up our bioinformatics pipeline, looking at dark genomic regions, and in particular, SMN1 and SMN2, and have had some success in running this quite rapidly. As you can imagine, identifications of patients with this condition, sequencing them, and classifying the SMN1 and SMN2 copy number, as well as any single nucleotide variants in this region as rapid as possible so they can get on therapy, is going to be very beneficial if they're presenting early on. We did a quick comparison between some of the tools that are available from ONT and some off-the-shelf bioinformatic mapping tools, and have identified a variety of methodologies that worked well for us. So our key success for whole genome in general has been that we've been able to surpass or match what we've had in short-read whole genome analysis for single nucleotide variants, indels, or structural variants. We've been able to do some benchmarking with some challenging medical relevant regions, looking at structural variants, and have been quite successful in detecting that. And what these are essentially are additional regions of the genome that essentially have been excluded from some of the classifications or variation benchmarks in the past. And this additional regions, of which include about 17,617 variants, we've had the opportunity to look at it on the ONT chemistry as well as with adaptive sampling. And on that, it runs 7-AS. That is our adaptive sampling benchmarking classification for these variants. And you can see just using adaptive sampling versus whole genome, we essentially reduced in half the amount of variants that were undetected originally just using whole genome. And then you can also appreciate the fact that our SMN1 region has increased substantially with adaptive sampling. But still, that is very difficult to do copy number variant detection in that region when you don't have reads that differentiate these two genes. So what we've essentially come up with is a kmer mapping approach, looking at SMN1 specific versus SMN2 specific regions. And this takes all reads out of a whole genome experiment. We know at least what 5 SNPs that classify SMN1 versus SMN2, and they're able to map those reads with Winomap and assign those to a particular loci. And looking at the combination of those kmers, we can count the number of SMN1s we have and the number of SMN2s we have. And this is just an example of the type of mapping quality we get from 3 copies of SMN1 versus 1 copy of SMN2. And this has allowed us to differentiate the deletions in this region as well as the copy number of SMN2. But what we're currently spending a lot of time is identifying how long reads do we need to be able to count all single nucleotide variants in every exon of SMN1. That is a 28,000 kb gene, so we assume we need around 30,000 read lengths in order to be able to cover those regions and call every single nucleotide variant. In our clinical lab, we're often asked the difficult questions because we're a reference laboratory. Diagnosing SMN1 based on spinal muscular atrophy based on an SMN1 deletion is quite simple. People can do this in a variety of different methodologies. The things they ask for are, I've got a patient with 1 deletion. Is this patient truly affected based on the clinical phenotypes? They look affected. Can you tell me whether or not there are any other variants in the exonic regions of that particular patient? And so we're asked to do that. So from our perspective, I think what we'll see quite rapidly is the use of this technology to cover the dark spaces. We also really anticipate the capacity to use this, as you've heard earlier, looking at expansion and methylation, as well as, I think, looking at a variety of different tissues. In particular, we heard a description of potentially mosaicism being involved, and I think there is some interest in looking at ONT and identifying whether or not the application is to detect mosaicism with a variety of different tissue sources. So I just wanted to thank you for the opportunity to speak and acknowledge quite honestly that we have a large group at Mayo Clinic. Steve Murphy, who's over here in the audience, is directing our advanced diagnostic laboratory where most of our ONT instrumentation sits. We wouldn't be capable of doing what we've done without their help. And we have a lot of the other individuals listed here who I really appreciate giving them the opportunity to work on this project. So thank you.